Good afternoon, future nurses. My name is Michael Ranyola, and I will be discussing the subtopic bases and nucleosides under the topic Nucleic Acid and Heredity. So before we start the discussion, please be advised, the information that you may read and listen might not exactly the same as how the book presented it. However, the content were still the same. Without any further ado, let's start. A nucleic acid is a linear polymer of monomers we call nucleotides. Now, sometimes when describing nucleic acid, instead of using the word nucleotides, we sometimes use nucleosides. But nucleosides and nucleotides are two different things. So in this lecture, we were going to discuss what nucleoside is. Let us first define what is nucleoside. When we say nucleoside, it is a structural subunit of nucleic acid, the heredity containing components of all living cells, consisting of a molecule of sugar linked to a nitrogen-containing organic ring compound. Like what I aforementioned, a nucleic acid is a linear polymer of monomers, and one of those monomers is what we call nucleoside. During polymerization, the molecules contained in nucleosides combine to form a large molecules that contain repeating structural unit, and that's when the nucleic acid form. So here's are some characteristics of nucleoside. Number 1. Nucleosides are glycosylamine that can be thought of as nucleotide without a phosphate group. When we say glycosylamine, it is a class of biochemical compounds consisting of glycosyl group, or also known as glycoside. All nucleosides is basically the nucleotides minus or without phosphate group, which means the difference between the two is nucleoside is absent of phosphate group while nucleotide has. Another characteristic of nucleoside, it is a unit that consists of a carbon-sugar molecule attached covalently via beta-glycosidic bond to a base. Third characteristic, nucleic acid contain nucleotides with two types. There are derivative of D-ribose, known as ribonucleosides, and 2'-deoxy-D-ribose, known as deoxyribonucleoside. Take note that ribonucleoside is a sugar molecule that only can be found in RNA. Similarly, deoxyribonucleoside can only be found in DNA. As you can see in this structure of two carbon sugar, in the left side corner is the sugar ribonucleoside, while on the opposite side is the sugar deoxyribonucleoside. In this particular nucleoside, the sugar is a ribose sugar, and what that means is the nucleoside will be found on RNA molecule. Now, what about the base? The base in this particular case is adenine, and the way we name this nucleoside is simply adenosine. Now, if this base is guanine, then we'd call it guanosine. If this is cytosine, we'd call it cytidine. And if this was uracil, we'd call it uridine. And if we take and remove this OH group, then we essentially change the sugar ribose into deoxyribose. And this will no longer be adenosine, it would be deoxyadenosine, and likewise, if this was deoxyribose, and guanosine would call it deoxyguanosine. If this is cytosine, we'd call it deoxycytidine. And if this is thymine, we'd call it thymidine. As you notice, the bond in a nucleoside that connects these two different molecules is always between carbon number 1. 1' on the sugar and 1 nitrogen found on the base. The bonds always takes place between carbon number 1 and nitrogen found on the base, and this bond called beta-glycosidic bond. Let's proceed for the fourth characteristics. In nucleosides, the anomeric carbon is linked through a beta-glycosidic bond to N9 of purine and N1 of pyrimidine. So let's first define what is purine and pyrimidine is. So purine is a heterocyclic aromatic organic compound that consists of two fused rings together. It is also water soluble. And pyrimidine is a heterocyclic aromatic organic compound that consists of a simple ring molecules. So this is the structure of purine and pyrimidine. As you can see, purine is placed on the left side corner, characterized for its two rings fused together, while on the opposite side is a pyrimidine that consists of simple ring molecules. So before we proceed to nucleobase or nitrogenous bases, let's recall the example of nucleosides. Under ribonucleoside, we have adenosine, guanosine, cytidine, and uridine. And under deoxyribonucleosides, we have deoxyadenosine, deoxyguanosine, deoxycytidine, and thymidine. Let's move on to nucleobase. So what are these nucleobases? Nucleobase, also known as nitrogenous base or simply bases, is a molecule that contains nitrogen and has the chemical properties of a base. Also, a nitrogen-containing biological compounds that form nucleosides and has the ability to form base pair and to stack one upon another leads directly to a long-chain helical structure such as ribonucleic acid and deoxyribonucleic acid. If you remember the fourth characteristic of nucleosides, I had already mentioned the word purine and pyrimidine. But in this case, in nucleobase, there are 5 nucleobases that are classified into 2 types: the purine bases and the pyrimidine bases. Under purine bases, there are 2 nucleobases named adenine and guanine, while cytosine and thymine and uracil are classified as pyrimidine bases. Remember that purine consists of 2 rings fused together and pyrimidine consists of simple ring molecule. So in this structure, adenine and guanine have a fused ring skeletal structure derived of purine, hence they are called purine bases. Similarly, the simple ring structure of cytosine, thymine, and uracil is derived of pyrimidine, so they are called pyrimidine bases. To form the helical structure of DNA and RNA, these 5 nucleobases are necessary. The nucleobases adenine is paired with thymine and guanine is paired with cytosine to form double helix that connects those strands compared to a rungs of a ladder in DNA structure. And the nucleobases adenine is paired with thymine and guanine is paired with uracil to form the RNA strand or structure. As you notice, the DNA and RNA were different in base pairing when it comes in the bases thymine and uracil. It is because the two are distinguished merely the presence or absence of a methyl group on the 5th carbon of these heterocyclic 6-membered rings. I think that this would be the end of this lesson. If you have questions, clarifications, violent reactions, feel free to ask. And I do hope that you have learned a lot. Thank you and have a nice day ahead.